Hi, I'm Liam, and in this video I'm going to be talking about the enzyme catalase and how it breaks down hydrogen peroxide into products of water and oxygen. In this lab, I tested the factor of different concentrations of an enzyme and how that affects reaction rate. The reaction rate is how fast the reaction is happening with the enzymes involved. Enzymes are macromolecules, normally proteins, that speed up chemical reactions by lowering activation energy barriers. To do this experiment, I diluted yeast, which contains the enzyme catalase, and water into 5 different concentrations: 0, 2.5, 5, 7, and 10 grams per liter. I dipped a filter paper into each of these solutions, placed them in the bottom of a test tube with hydrogen peroxide, and measured how long it takes for each filter paper to float to the top. The reason this filter paper disc floats to the top is because the catalase, which is found in the yeast solution, is an enzyme with an active site that bonds to the hydrogen peroxide, which is a substrate, and it breaks it down, which releases water and oxygen. The substrate is like the ingredients in cooking. Without a substrate that fits into the active site, the reaction will not be able to occur, just like how you can't cook without any type of ingredients. And in the products, the water doesn't really affect the experiment, besides its being combining with the hydrogen peroxide. However, when oxygen, the other product, is released, it forms bubbles, and some of those bubbles get caught in the filter paper. When enough does, the filter paper rises to the top. Here is the data from my experiment. As you can see, the higher the concentration of the yeast solution, which contains the enzyme catalase, the faster the reaction rate. This is measured by floats per second in the graph. More enzymes allow for the enzymes to react quicker and more efficiently with the substrates binding to the active site and releasing the results of water and oxygen. To go back to the cooking analogy, more chefs means more products in less time. In the data, there is a plateau at a certain point on the graph when the concentration is fully saturated or close to fully saturated. This is because increasing the concentration of yeast or increasing the proper proportion of catalase enzymes eventually doesn't matter, since the enzymes have already broken down so much of the hydrogen peroxide into water and oxygen that it won't change the results at a certain point in time. More saturation of enzymes means the proportion of enzymes is greater, which translates into a higher reaction rate, which is shown by the graph. As mentioned earlier, in this experiment, the concentration of yeast solution was what was being changed, or the independent variable. In a more molecular context, this is an increase in the proportion of catalase contained in the yeast. The higher the concentration of the yeast, or the higher the amount of available enzymes, the higher the amount of floats per second there were. The floats per second, in basic terms, is the reaction rate, and how fast it takes for the enzymes to catalase the reaction, which in this case was turning the hydrogen peroxide into oxygen and water. As mentioned earlier, in this experiment, the concentration of yeast solution was what was being changed, or the independent variable. In a more molecular context, this is the increase in the proportion of catalase contained in the yeast. The higher the concentration of the yeast, which could also be said as the higher the amount of available enzymes, the higher the amount of floats per second there were. The floats per second, in basic terms, tracks the reaction rate and how fast it takes for the enzymes to catalyze the reaction, which in this case was turning the hydrogen peroxide into oxygen and water. Another thing to consider in these processes is that when less active sites are available, which would be the case if the concentration of the yeast solution was diluted, the substrates would have fewer opportunities to bind to the active sites, and therefore the processes would slow down. So here I'm going to explain what's really going on in the experiment in a more molecular level and a more close-up level with toothpicks. So here we have what would be a more diluted solution with water, which is what I'm testing for, like the different dilutions and concentrations. So here we have a more diluted, and So here my hands are the substrates, and then these would be the active sites waiting for the substrates to bind onto, and the enzymes can catalyze the reaction. So basically here are the substrates. They're moving and they're looking around, but there's a lot of open space here, and it's harder for them to run into an active site. There's less active sites here. And then maybe once in a while they'll run into one, and boom, a reaction happens. But then once a reaction more substrates are going around, and then maybe, another one will hit another active site. And then it breaks. And that's how a reaction happens when it's more diluted. There's less, and then therefore there's more space for the enzymes, and then there's less substrates and then less active sites. And therefore it's a lot slower, and the reactions don't happen as fast. But here is what happens when we have like a fully concentrated solution. So here we have, again, my hands are the substrates, and then these are all the active sites. When my hands are moving around, easily, boom, I run into active site here. The substrate, boom, reaction happens. Boom, I'm moving around. Another substrate hits binds with the active site. Boom, breaks. Oh, another one right here. See, it's more active sites are available for the substrates in a closer proximity, and this allows for a quicker reaction, and this is what happens more at a molecular level. This is why the more diluted and less saturated solutions had a slower time flowing to the top. However, the more saturated the solution was, the more catalase enzymes were available to catalyze the reactions. Enzymes tend to work better in more saturated solutions. This can be explained simply. When there is no water diluting the concentration of enzymes, there's nothing affecting the proportion of enzymes. So therefore, it's just an enzyme that doesn't need to go through any difficulty binding the substrate to the active site, since there's so many available. Hopefully this helped you understand the experiment a little bit better, and I thank you for watching.